Hey there, I'm Richa and welcome to iBiologist. Today we're gonna learn about endoplasmic reticulum, which is abbreviated as ER. ER is the cell organelle which is absent in almost all the prokaryotic cells, and it is found in all the eukaryotic cells except germinal cells and the mature mammalian RBC. Before we are going to the structure of the ER, it is very important to know that there are two types of ER, rough endoplasmic reticulum and smooth endoplasmic reticulum, which is abbreviated as RER and SER respectively. There are three elements of ER. First, which is cisternae. Second is vesicles, and third one is the tubules. Cisternae is the stag-like structure which is originated from the nucleus membrane, and it is studded with the ribosomes. Ribosomes are attached by a protein, ribophore. Due to the presence of this ribosome, the surface of the cisternae is rough, and that is why it is called as rough endoplasmic reticulum. As we see, the second element of the ER is vesicles. Vesicles are 50 to 500 micrometer in diameter, and it is closer to the cisternae, which is nearby the nuclear membrane. The third element of the ER is tubules. Tubules are branched without ribosome. It is closer to the plasma membrane or the cell membrane, which is 50 to 100 micrometer in the lumen, and it is closer to the plasma membrane. ER in the muscles is known as sarcoplasmic reticulum. Vesicles are free of ribosome as like tubules. If vesicles formed RER it has ribosome, and if it is formed from SER it will not have ribosome. Smooth endoplasmic reticulum are tubular, whereas rough endoplasmic reticulum are mentioned as cisternae. Before the video ends, let's differentiate the RER and the SER. As its name shows, RER is rough endoplasmic reticulum and SER as the smooth endoplasmic reticulum. Cisternae is RER, whereas tubules are SER. RER is closer to the nuclear membrane, whereas SER is closer to the plasma membrane. There is ribosome present on the surface of RER, where SER has no ribosomes on the surface. RER helps in the protein synthesis, whereas ER helps in lipid synthesis or its transport. Rough endoplasmic reticulum transport of nuclear material from the nucleus to other organelles. SER forms desmotubules strands of endoplasmic reticulum which runs through plasmodesmoto sarcoplasmic reticulum, which turns into the SER. Further are desmotubules, and the cytoplasmic connection between them is known as plasmodesmo, which is further connected into the sarcoplasmic reticulum. Now we will see the location of RER and SER, or the cell who have more amount of this. So, as we see, the function of rough ER is protein synthesis. The cells that secrete the protein would have more rough ER, right? So, here are the examples which have the larger amount of rough ER. Fibroblasts are the cells who produce and secrete a larger amount of rough ER. And the B cells, which are responsible for protein synthesis, which are secrete insulin as protein, so they would have more rough ER. As the same way, the function of smooth ER is lipid synthesis. So the cells that synthesize lipid would have more amount of smooth ER. Adipocytes and liver cells which produces more smooth ER, and as the steroids are lipids, steroid synthesis cell like follicular cells and Leydig cells have more SER. As the follicular cell secretes steroid hormones like estrogen and progesterone, while Leydig cells are synthesize male steroid hormones like testosterone. So both of them, follicular cells and Leydig cells, will have more smooth ER. Let's see the functions of smooth ER. As we see, the sarcoplasmic reticulum is smooth ER. The functions of this will go under smooth ER. So, the conduction of motor impulse in muscles and the removal of lactic acids in joints are the functions of sarcoplasmic reticulum. It is also helps in detoxification of carcinogenic substance and the glycogenolysis in liver cells.